It is basically a combination of multiple techniques which are simultaneously analyze multiple measurements or individual object under investigations. But when we have multiple options for analysis, then we have a very critical situation how to decide which technique will be applied on which scenarios. Hence, we can we face three major questions during analysis. First is, can the variables be divided into independent and dependent classifications based on some theory? It means uh, is there any dependency between the variables? Suppose one is dependent variable and other variables are independent variables which are affecting our dependent variable. Second questions we face, if there is any dependency between the variables, then how many variables are treated as dependent in single analysis? Because dependent variable is our the most critical variable. So the number of dependent variable in the study is also very crucial. And the third problem is how are the variables, both dependent and independent variables, are measured on which scale? We all know, I think you all of know that there are various scales of measurement for variables such as nominal scale, ordinal scale, ratio scale, interval scale. So on the basis of these three questions, we select which multivariate techniques should be applied. So here is a flow chart for the to understand the mechanism of multivariate analysis. If we see here, uh, 1 minute. If we see here, so first question we have, is there any dependency between the variables or not? So if we see in this flow chart, we have first question, are some variables dependent or not? If there is some dependency, we can use dependence methods. If there's no dependency between the variables, it means all the variables are independent to each other. Then we use interdependence methods. If we see under dependence methods, then our second question arises, how many variables are dependent? If one variable is dependent or more than one variables. Then our third question arises, if one variable is dependent, it is measured on metric scale or not? We are going through three questions one by one. First question we face, is there any dependence between the variables? If there is any dependency, we check how many variables are dependent. If one variable is dependent, then we check it is measured on which scale. If it is measured on metric scale, we use multiple regression. Otherwise, we use multiple discriminant analysis. Similarly, if we have more than one dependent variable and it is measured on metric scale, then we can use multivariate analysis of variance, multivariate ANOVA. If not, we can use canonical analysis. Similarly, if there is no dependency between the variables, it means all the variables are independent to each other. In that case, we check if our independent variables are metric or not, measured on metric scale or not. If they are measured on metric scale, then we can use factor analysis, cluster analysis, and metric multidimensional analysis. If not, then we can go to non-metric MDS, latent structure analysis, and etc. So here we will today discuss about two multivariate analysis techniques. First one is PCA, principal component analysis, and the second one is cluster analysis. Now, what is PCA, principal component analysis? We all know that uh, if you have taken lecture on regression analysis, the basic assumption of regression analysis is that there should not be any multicollinearity between the variables. It means the variables should not be highly correlated. But in the field of agriculture, for example, suppose we have plants related traits, they are mostly correlated with each other, mostly highly correlated with each other. Suppose plant height, number of branches, number of secondary branches, and etc. These are plant traits. Most commonly, these are highly correlated. In this case, we cannot apply regression analysis on these variables due to assumption of non-multicollinearity. To overcome this situation, we use principal component analysis. The core of principal component analysis is that among a set of multiple correlated variables, we transform them into new sets of uncorrelated variables. These new set of uncorrelated variables are called principal components. These basically component we derive principal components are the linear combination of original variables. Suppose we have five variables x1, x2, x3, and x5. Then we extract different principal components from these variables. The obtained principal components will be a linear combination of these five variables. And next important point is the obtained principal components are fully uncorrelated with each other. Then, why it is necessary in agriculture? In modern agriculture, we have faced most of the data set like this. Suppose high throughput phenotyping, drone and satellite capture hundred of spectral bands per plant. In genomics, we have genotyping by sequencing yield tens of thousands of SNPs. In soil science, we have 20 plus chemical properties which are all correlated with each other. In weather data, daily temperature, rainfall, humidity, these are all highly correlated with each other. We can use PCA in these scenarios. Now, suppose we have a problem statement. Suppose we have 50 variables. We cannot visualize the data in 50 dimensional space. In this case, we use principal component analysis. Basically, principal component analysis is a data reduction technique. It reduces the many variables into few principal components. Then what is the solution? PCA transforms these correlated variables. Suppose we have 50 correlated variables, then PCA will transform these correlated variable into smaller set of uncorrelated variables called principal components while retaining as much variation as possible. What are the mathematics? It is a basic mathematics behind the PCA. I have already stated that PCA is a linear combinations of your variables. Now, first step is standardization because suppose we have 50 variables, independent variables, they can be measured on different units. Suppose, for example, agriculture variables are measured in different units, kg per hectare, pH, degree Celsius. If we do not standardize this variable, it will not be feasible to analyze this because suppose we have yield data. Yield data varies from higher values. Suppose yield is 5,000 kg per hectare, while pH value is 6.5. If we numerically compare both values, 5,000, 6.5, there's large variation between the variables data. So we reduce it to on the scale of 0 to 1. That is called standardization or normalization of data. We do normalization of data by using this formula z is equal to x minus x bar divided by standard deviation. After standardization, your data is reduced between 0 and 1. Then second step, calculation of covariance matrix. We calculate how every variable varies with every other variable. Basically, we calculate the covariance between all the variables under study. We face two types of scenarios. First is positive covariance. Suppose more nitrogen means more chlorophyll. Second type of negative covariance, where there is negative relation between the variables. Suppose high temperature and lower soil moisture. If there is high temperature in the atmosphere, soil moisture will be lower. Similar like correlation. Third is eigen decomposition. Third step, we calculate eigenvectors and eigenvalues of variables. Eigenvalues show the how much variance is captured by your each principal components, and eigenvectors show how much tells us the contribution of each original variable on the principal component. Up, Now we, after these mathematical uh, observations, now we have the outputs. In PCA, principal component analysis, first output we have scree plot, which is a bar plot showing eigenvalues. On the basis of this scree plot, we decide how many principal components we have to retain in our analysis. For example, in a wheat trial, suppose 10 agronomic traits are there. It means when we perform PCA, we will have 10 principal components. For each variable, a separate principal component is generated. If we have 10 variables under a study, then we have 10 principal components generated. But we cannot select all the principal components. We select only those principal components which have higher variance. For example, in a wheat trial with 10 agronomic traits, suppose PC1 has 40% of variance of your data and PC2 has 25% of variance of your data, PC3 has 12% of data, and rest are noise. If we combine the variance of all PC1, PC2, and PC3, we have almost 78, 77, 78% of overall variance. Now we can select PC1, PC2, or PC3, maximum up to PC3, because rest are the noise. So out of 10 agronomic traits, we kept only two or three principal components. That in this way we reduce the dimensionality of data. Second is loadings. I have already explained in previous slide what is loading. Loading is basically correlation coefficients. Suppose we have found this type of, suppose we have four variables: grain yield, plant height, days to maturity, and protein content. And we found two principal components, PC1 and PC2. Now, here 0.85 shows that grain yield variable is the correlation between the grain yield variable and first principal component. And 0.10 value shows that the correlation between grain yield variable and second principal component. The variable which has higher loadings, that variable go to the that principal component. For example, here grain yield will go to principal component 1 because it shows high correlation with principal component 1. Similarly, plant height will go to principal component PC1 because it has higher correlation 0.80 in PC1. Days to maturity will go to principal component 2 because it has higher correlation with PC2. Similarly, protein content has a higher correlation with PC1, but it is negative. So we have to interpret in that way. Grain yield, plant height, and protein content are going to principal component 1, but protein content is showing negative correlation with first component. And now last is the transformed value of each observation. After uh, conducting PCA, we get the new principal component scores of each principal component. And on the basis of this, we can go to for other statistical technique like clustering, outlier detection, and regression. So basically, we have what we have done here. We have, suppose for this example, we have 10 agronomic traits. On the basis of those 10 agronomic traits, we selected two principal components. Now we get the PC score for both the principal components. And now we have two variables, PC1 and PC2, which are fully uncorrelated with each other. Now we can conduct regression analysis because the assumption of regression is there should be no correlation between the variables. Earlier, the 10 agronomic traits are highly correlated with each other, but newly correlated PC1 and PC2 are uncorrelated. Hence, we can apply regression technique on this principal components. Now, how can we apply this in agriculture? First is genotype by environment interaction. One of the biggest challenge of plant breeding is which variety perform best across different locations. Suppose we have 20 wheat genotypes grown in 10 different environments. Then how can we analyze this data? Suppose we have 10 environments and 20 wheat genotypes grown in all these 10 different environments. We use PCA. You have a surely known study AMI analysis, AMI stability analysis, which is based on principal component analysis. It reduces the data and generate principal components. On the basis of these principal components, we found which uh, genotype is stable in across the all the environments. Second application may be hyperspectral imaging. Drones capture reflectance across 200 plus wavelength, and all the wavelength are numerical numbers, so they will be highly correlated with each other. In this case, we cannot direct apply any statistical technique like regression. Hence, in this case, we use PCA to reduce the correlation between the variables and get the reduced data. Third application may be soil fertility mapping, where we have suppose we have 15 chemical properties of soil, and all the soil properties are correlated with each other. Then we can use PCM. Fourth is outlier detection in farm sensors. Suppose our dairy farm has 100 sensors per cow, milk yield, rumination time steps, temperature. Then in this case, we cannot analyze 100 variables simultaneously. So we reduce the data using PCA. These are some basic concept of principal component analysis, but it has some limitations also because there are some assumptions based on all the statistical technique. First one is linearity. PCA only captures linear relationship. Earlier starting, I have said that PCA is a linear combination of your independent variables. Hence, there should be some linear relationship between the variables. If there is non-linear relationship between the variables, PCA will not capture it. Suppose, for example, yield and temperature. Initially, when temperature is low, yield is low. As the temperature increases, yield also increases. After a saturated temperature increases, the yield also started to decline. Then we have a U-shaped relationship between yield and temperature. In this case, PCA will not capture correctly this data. Second is sensitivity to outliers. Sim, PCA is highly affected by the outliers. Hence, we have to first clean our data before analyzing PCM. Third is interpretability, because in principal component analysis, we can get multiple principal components. So we have to decide which principal component should be retained in the study and which should not be retained. Suppose you have selected four principal components out of 100 variables. Then if we plot these data, we can plot only highest two principal component, PC1 and PC2, due to dimension reduction. Fourth is skill dependency. If we do not standardize our data, then it will also create some fuzziness in your study. These are the some limitations of principal components. So make sure you first we check in when we start this principal component analysis. First we check the linearity between the data. If there is linear relationship between the data, then we check correlation between the data. If there is high correlation between the data, it is most suitable for principal component analysis. Now, second is cluster analysis. Before going to cluster analysis, I want to uh, some R. analysis on, I think all of you are aware why with R. So suppose I have generated this data set in randomly. Suppose I have generated this crop data where I have selected first one is variety. First variable name is variety. I have generated a variety data of 10 varieties. Second is yield variable. I have written here mean of this 5 and standard deviation 0.8. Second variable I have taken is height with mean height of 120 centimeter and 15 centimeter of standard deviation. Then I have taken the variable maturity with 100 as 100 days as mean maturity days and 8 is standard deviation. Then I have taken protein, then lodging and disease. Now we have six types of variables in this study. First one is yield. Variety is your categorical data, which has 1 to 10, V1, V2, V3, and V10. Second one is height. Third is maturity. Fourth is protein. Fifth is lodging. And sixth is disease. Now, if we run this data, for example, I want to see this automatic generated data. I have used head crop data. This is your generated data. If you want to see all the 10 fields, suppose I am using view function for generated crop data. This is your automatic generated data, where first one is variety. We have 10 varieties, V1, V2 up to V10, then yield, height, maturity, protein, lodging, and disease. On the basis of input I have given, this data is generated. Now I want to go for principal component analysis on this data set. If we see here in this data set, first one is our variety. So we have to exclude this variable first because it is a categorical data. It is not a numerical data. It is not measured on metric scale. V1, V2 are categories. So first we have to exclude this variable, variety named variable. So what we have done here, we have created a new variable traits. Here traits, I have used crop data, bracket open, and I have written -1. -1 means I have removed the first column of crop data. This is your crop data, and first column is variety. I have created a new variable named traits where I have used -1 function to remove the first column of your data set. Now, if I see what is under your traits, newly created traits, sorry, I have to run first. No, the variety column has been removed from this data set. Now we have a new variable called trait, which has all the six traits of your data. Then our first mathematical intuition is we have to standardize your data set. So standardization means mean is 0 and standard deviation is 1. So we use standardize the traits mean is equal to 0 and as standard deviation 1. We have created a new variable traits underscore scaled, and we have scaled this traits previously created trait data set. I'm running this trait scale. Now if I want to see what are the look of variables after standardization, I'm checking it. Now, this is your standardized data. Earlier we have this data set, yield, height, maturity, protein, lodging, and disease. Now we have standardized it using standardization formula z is equal to x minus x bar divided by sigma. Now we got this standardized data set. Okay. First step is complete standardization of your data set. Now we run the principal component analysis. Here, for principal component analysis, we have direct function, inbuilt function, PR, COMP. So, suppose I am using PRCOMP function and using first I have to insert the variable, standardized variable. So we have standardized the variables in traits and scaled, traits underscore scaled. So we have written here traits underscore scaled, then scale true and center true we have selected. If we run this, this is your output. Earlier I have said if you have 10 variables, 10 principal components will be generated. Here we have 6 variables under study. That's why 6 principal components are generated here. Okay. Now we save this score under this variable PCA result. So I have created variable PCA underscore result and save this result of principal and component analysis under this variable. Then we have found the summary of principal component analysis result. This is your summary. We have 6 principal components. Now, if we see here proportion of variance, first principal component has 43% variation of your data. Second principal component has 24.15% variation of your data. Third principal component has 14.5%. Fourth has 12.13%. Similarly, 4% and 2% almost. This is your cumulative proportion. First principal components explain 43% variability of your data. First two principal component cumulatively explain 67% variability of your data. First three principal components explains 81.75% variability of data and so on. Now we have to calculate the variance explained by each principal component and we have to plot it. I have already told you about scree plot, which is basically a bar diagram which shows the percentage of variability explained by each principal component. To plot this, we use library facto extra. So if you have not installed this package, you have first install under under package install facto extra, click on install. Your package will be installed in your system. Then we call this package by using library function, library facto extra. I'm running this command, already library installed. Now I'm using f which underscore eig function for visualizing our data principal components. Here, suppose if you want to see the it is used for visualization of eigenvalues or variance of your principal components. The syntax of this function is like this: fviz underscore eig. First, we have to give x. X is your PCA results of principal components. So that's why I have written here PC underscore results. Then, if I want to add labels in this bar diagram, I am giving add labels is equal to true. By default, it is false. It means it will not give any label to your graph. If I want to add any label in my graph, I am using add label equal to true. Then I am giving y-axis value 0 to 50. And I have given title here, script plot for variance explained by principal components. If I run this command, I will get this script plot here. I have already given it title script plot variance explained by principal components. And on the basis of my score, principal component score, which is shown here, you can see here first principal component has 43.09% variability. It is shown here. Second principal component has 24.15. It is shown here. And so on. All the six principal components variance is shown in this script plot. Now, second result we found here is biplot. Biplot is basically the graph between your first two principal components because the first two principal component explain the maximum variability of your data set. If we here see that first principal component has 43% variability and second principal component has 24% of variability of data. Again, we use fwiz underscore pca by plot function. Then I have first inserted my PC results here. If you go want to find the syntax of this function, you can here search. This is the syntax of your biplot. First, I have to explain X, which is your scores. I have written here PC results. Then you can use most of these subsections of this function. I am using some, suppose repel function. Repel function used for avoid text overlapping. Second, I am using colors for variance and color for individuals, varieties by blue and red colors. And I am giving title to this graph, PCA by plot of crop traits. If I run this, I will have this biplot here. Here, first PC principal component, which has 43% variability, is shown on x-axis. Second principal component, which has 24% variability, it is shown on y-axis. And all the six traits are given here with the all 10 genotypes. Then I want to find which principal component contribution of each trait in principal component 1 and principal component 2. Basically, I want to find loadings, which are correlation coefficient I have discussed earlier. For this, I have again created a new variables loading, and under load, suppose I'm writing here loadings. Now, under your PC results, there are already various results. So when I read PC results and give dollar sign, we have all these inbuilt outputs here: standard deviation, rotation, center scale, and X. So I am using here rotation. I have run here. Then now I have created a new variable loadings. I want to this print. So I have given print command. Now we have output factor loadings of each principal component. For example, yield has high correlation of with principal component 1, which is 0.514 in comparison to PC2. Height variable has high correlation with PC1, again Maturity has high correlation with PC1 again, but it is in negative direction. Protein has again high correlation with principal component 1. Lodging has high correlation with PC2, and disease has high correlation with PC2. It means in PC1 we have four variables: yield, height, maturity, and protein. And in PC2 we have two variables: lodging and disease. This is the concept of principal component analysis. We have 10 variables and we have reduced it into two variables which are now not correlated with each other. Earlier, all the 10 variables are highly correlated with each other, but now the newly created PC1 and PC2 are not correlated with each other. These are uncorrelated. So we can use any interdependence technique on this data set. Now, this is principal component analysis. Now we have second topic is cluster analysis. In cluster analysis, I have earlier said that cluster analysis is also a multivariate techniques where we have not any correlation between the variables when there is no dependent or independent variables under the study. Okay. Cluster analysis is a multivariate technique whose main purpose is grouping of objects based on your characteristics they possess. Basically, it is a classification technique which is used to classify the data into different groups on the basis of similarity and distance. Okay, these cluster analysis classify objects on a set of user selected characteristics called the clustering variable, and these resulting group of objects are called clusters. For a successful classification of objects, there should be high level of homogeneity within clusters and high level of heterogeneity between the clusters. Um, why can we are using cluster analysis in agriculture? Again, the same problem. Agriculture generate data on multiple scales. For example, individual plants, field, farms, and regions. Okay. Sometimes we have predefined classification. Sometimes we do not have predefined classification. If we have predefined classification available, we can use discriminant analysis or other classification techniques. But if we do not have predefined classification, we have unclassified data, then we can use cluster analysis for classification and the grouping of your data set. In which scenarios we can use, for example, precision agriculture, where we want to divide multiple management zones on the basis of fertilizer and irrigation. In plant breeding, we can group genotypes on their performance across the environments by using their morphological traits. In livestock farming, we can use animal health data for clustering. In crop classification, we can identify multiple crops on the basis of satellite imagery without ground truth labels. In soil science, we can define soil functional classes based on physical and chemical properties. Now, these are already I've discussed applications in agriculture. How can we apply? These are some uh, different scenarios. For example, precision agriculture. I have earlier said that we can classify multiple management zones based on fertilizers, seed and irrigations. Suppose we have data of soil electrical conductivity, historical yields, elevation, and satellite imagery. On the basis of this data set, we can identify the multiple groups across the field. Similarly, in plant breeding, a breeder evaluates 200 wheat lines for 15 agronomic traits. Now, we have 200 wheat lines. We do not have any prior idea about which uh, uh, which wheat lines are similar and which are not similar with each other. Then we can use cluster analysis to identify the similar genotypes and not similar genotypes. All the similar genotypes will be clustered into one group, and the which are genotypes are not similar, they will be classified into another cluster. Similarly, crop classification from satellite imagery and livestock behavior clustering. These are some examples. Now we have how to we conduct the cluster analysis. First step is formulation of your problem. Earlier I have already said that we have unclassified data. We can use cluster analysis when we have unclassified data. We have no classification about the data set. Okay. Then how can we use cluster analysis? On the basis of some past research, theory, and consideration of hypothesis, we are assuming something. If cluster analysis is used as an exploratory research, the researcher naturally exercises her judgment and intuition. Okay, it means we have no prior knowledge about your data set. We are we have no labels on your data set. These are all totally unclassified. Then we have to find which data is similar and which is not. Second step is distance measure. If we want to find which data is similar and not, we have to put some mathematical measure between the data set. For keep, keeping this, we have used different distance measures such as Euclidean distance, Manhattan distance, Malnovich distance. All of you are aware about these. These distance measure is used to find the distance between your data set. If the distance is less, then the data set will be called similar. If distance is very high, the data will not be called as similar data set. Okay. Object with smaller distance between them are more similar to each other than those are at larger distance. Third step is select a clustering procedure. There are various clustering procedures for hierarchical clustering like linkage method, average method, variance method, centroid method, word method. These are some clustering process. So we can select any of these in for analyzing our data set. Fourth is decision on the number of clusters. Basically, when we conduct cluster analysis, because it is unclassified, we can have multiple number of clusters. So the main question arises, how how many numbers of clusters should be taken into consideration? For this, we have no hard and fast rule, but some techniques are created to identify the number of optimized clusters. Some are elbow methods, silhouette analysis, and gap statistics. These are used for finding the optimum number of clusters. Dendrogram is also used to identify the number of clusters. And fourth one is access reliability and validity because we have unclassified data, and on the basis of cluster analysis, we are grouping them in a cluster. So we have to check the reliability and validity of your results. For this, the, we can use various techniques. For example, perform cluster analysis on the same data using different distance measures. I have earlier said that there are various distance measures. For example, Euclidean distance, Manhattan distance, Malnovich distance. So we can change the different distance measures and find that if there is a similarity in the results or not. If the results are similar across the all the distance measures, then our answers are stable, our solutions are stable. Similarly, we can use different clustering method also and compare the result. Third is we can split our data into two groups. In the We are now discussing about artificial intelligence, deep learning, and machine learning. The basic concept of machine learning, deep learning is that we have a large data set. So what we can do with this data set? We uh, split this data set into two or three categories. First one is testing data set, second is training data set. We the ratios can be 20 to 80%. So for example, 80% data should be kept as training data set and 20% data is be kept as testing data set. We train our model with 80% data and we test with the rest 20%. Similarly, we can use this in cluster analysis also. We can split the data into two halves, perform clustering separately on each half and compare the results. Then we can use some checks also. We can delete some variables randomly and then perform cluster analysis. Suppose you have 10 variables and so we suppose you deleted one variable. See, it means you have conducted cluster analysis on 10 variables, then conducted on 9 variables, then 8 variables. Is there any similarity between the result or not? If there is any similarity, it means the variables you have deleted from your data set has no contribution in the clustering. If your results are drastically changed, it means the variables you have removed from your data set has the higher contribution while clustering. Okay, so now we can go to the analysis of clustering. We are using the same data set which we have generated. This is crop data. We are using the same data set for clustering also. So for clustering, we use, I have already said that this is your data set. So suppose you have six variables, yield, height, maturity, protein, lodging, and disease. Now you have 10 observations, V1, V2, V3, and V10. In PCA, we are reducing the number of variables. Okay. we have we had six variables and we had reduced it into two principal components. Okay, now we are in cluster analysis, we classify our data set and classification is done on the responses. Here we have 10 different responses. Okay, there's some difference between both the analysis. In PCA, we reduce the variables, not the observations. We do not reduce the observation. We only reduce your variables. Okay. But in clustering, we do not make grouping of your variables. We group the number of observations on the basis of these variable. Okay. So it means when we get your result, we will have the groupings of V1, V2, V3, and V10, not the grouping of yield, height, maturity, and protein. For example, in decimal component analysis, we have yield, height, maturity, and protein in PC1, lodging and disease in PC2. Okay, here variables are classified into different principal components. Okay, but in clustering analysis, the number of observations will be classified into different groups. Okay, so first we calculate the distance matrix between the variables, where between the your observations. We have 10 observation, V1, V2, and up to V10. So first we calculate the distance between V1 and V2, V1 and V3, V1 and V4, and so on, like this. So first we calculate distance matrix. Now for distance matrix, we use dist function. If you can go in here, okay, not showing. Okay, so we are creating a distance matrix here using dist function. So already we have scaled our data set here using norm standardization. So we are using this data set into for calculating the distance between the variables because it is already ranges between, it has already mean zero and variance one. It is normalized. So we are using this data set. So I have inserted trait underscore scale and which distance measure I am using it is Euclidean. You can use other also. Suppose Manhattan you can use, Malnovus you can use. Okay. I am using here Euclidean basis, which is basic uh, distance measure of in cluster analysis. Okay. So I have run here this. If you want to see distance matrix, click and run. This is your distance matrix. It means V1 and V2 has the distance of 3 point. V1 and V3 has the distance of 2.64. V1 and V4 has the distance of 3.14. This is your distance matrix. We have 10 observations. That's why we have here 1 to 9 in columns and 2 to 10 in rows. Now, we want to perform hierarchical clustering here. So we are using hclust function here. Hclust function. This is used for hierarchical clustering. This is syntax of hclust. Hclust. First, we have to give here D. D is distance matrix. We have already calculated distance matrix in the previous command. So we have written here dist underscore matrix. Now, which method I am using for clustering? I have already said there are multiple clustering methods, average method, linkage method, centroid method, ward method, ward d square method. Multiple methods are given. So I'm using here ward d square method. Okay. You can also find a multiple ward point, ward dot d, ward dot d2, single, complete, average, McVitie, median, centroid. Okay. You can write any of these method here, clustering method here. I'm using a word point D2 because it is most commonly used. I've run this. Now, if you run HC, it providing, I'm using clustering method word point D2. Distance is Euclidean and number of objects are 10. We have 10 observations. Now, I want to plot dendrogram for this cluster analysis. So I'm using plot function. I have already performed clustering. I'm using that variable fc into here. Then I am giving title dendrogram for crop varieties. I am defining the x-axis using x-labs of section, here varieties, and I am giving x-axis value 0.8. If I run this command, I will get dendrogram here. You can modify it by using various inbuilt techniques by using ggplot library package. This is a basic dendrogram. Here we can see that on the x-axis there are varieties. So we have 10 varieties. So these are 10 varieties given here. On the y-axis, it is showing height. Height is basically the distance you have calculated already. Okay. Now, if you see here, the maximum distance is more than 6. Okay. Uh, how to identify the number of clusters on the basis of your dendrogram? Suppose initially, we have only one cluster which has all the 10 genotypes. So if we are at this line we have all the 10 genotypes under one group, one cluster. Okay. Further, it breaks into two parts. First one is here, and second one is here. As we have a break in your horizontal line, the number of clusters are begin to increase. Initially, we have all one cluster. Now, as this horizontal line breaks, we have two clusters. In one cluster, first cluster, we have four genotypes, 3, 6, 1, and 5. And in the second cluster, we have remaining six genotypes. Okay. Now we if go down to or near to the low height, low distance, we again have a break here. Okay. At this point, we have again a break. It means the number of clusters will be increased at this point. Already we have this one cluster. Now the second cluster break into two clusters again. In the second cluster, we have 2, 9, 7, and 4. And the third cluster, we have 8 and 10. Okay. Similarly, if we go down, then we have again a break here. Okay. At this point, we have again a break. It means number of cluster again increase at this point. One cluster having 2 and 9 genotypes, and second cluster has 4 and 7 genotypes. Okay. So basically, if we see here, we have one, two, three, four, five clusters here. Okay. But we cannot use all the five clusters. Okay. So we have to find the optimum number of clusters. Okay. So how we define the optimum number of clusters? Okay. One minute. Cut the dendrogram to obtain. Suppose we are want to use three clusters. Okay. So I'm creating a variable clusters and using cut tree function where I use clustering variable HC and k is equal to 3. Okay, because I want to obtain three clusters. If I'm running this, now the genotypes are divided into three categories. First genotype is in first category, first cluster. Second genotype is in second cluster. Third genotype is in first cluster. Fourth genotype is in second cluster, and so on. If I increase the number of clusters 4 here, and then now we have the four cluster classification. Okay. Suppose I am reducing it to two clusters. Now my data is divided into two clusters only. Okay. So you can decide how many number of clusters do you want. Okay. By seeing this dendrogram. Okay. By seeing this dendrogram, you can decide how many numbers of clusters you want in your analysis. So I am again writing it here 3, running the command. Now I am at cluster assignment to the original data. Okay. So I'm using crop data underscore cluster. Crop data, the initial data we have. We have assigned the cluster to the original crop data here. Okay. By using this function crop data dollar cluster equal assigned to factor under bracket clusters. Okay. Now, if you want to print this, this is your cluster assignment to. Initially, if we see here, only the number of cluster grouping is given here, but not the name of varieties. So I have assigned this result into our initial data, crop data. So now we have the result, variety and cluster wise. Okay. First variety is given to first cluster, second variety is gone to second cluster, third variety is gone to first cluster, and so on. Now, if you want to extract PCA score, visualize cluster using PCA score, we can also combine both the techniques also. So suppose I am creating a new variable called score and I am assigning it PCA results in creating a data frame of your PCA results, which are earlier we have obtained. For example, PCA results, if I click here, PCA results I have created using PRCOMP command. I have calculated PC results here. I am using these PC results to create a data frame as a new data frame name at scores. I have run this command. Okay. Now I want to assigning cluster classification into the principal components by using this command. Okay. I have assigned the cluster classification into principal components. Now if I Now I'm using ggplot2 library for visualization. For visualization, ggplot2 is used in R. So I'm using this package. I have already installed here. I have initiated the library, initiate the package using library function. Now I have score using ggplot function where I have used my scores, access PC1, PC2 color, all I have written here, and I have run this command. This is your PC scores by using cluster analysis. On the x-axis, we have principal component 1, and on the y-axis, we have principal component 2. And the varieties are shown by using their cluster classification. Here, we can see that V5, V1, V3, and V6 are on the right side of first principal component, but are the first cluster. V8 and V10 genotypes in the third cluster, but in the negative direction of first principal component, and V2, V9, V4, V7 are in the second cluster. Okay. Now we can characterize each cluster by trait means also. So we have created a cluster summary here by using aggregate function. If I run, this is your cluster summary. Okay. We have obtained three clusters. This is your first cluster. First cluster has mean yield of 5.575 ton per hectare, average height of 130 centimeter, 91 average maturity days, okay, and so on. Similarly, in the second cluster, we have average yield of 4.9 tons per hectare, and average height of plant is 126 centimeter. Similarly, the genotypes which are clustered into third cluster have average yield of 4.3 ton per hectare and height of 102 centimeter. This is your means of different clusters. Okay. So this is your basic soft cluster analysis. You can use more inbuilt functions of R to more visualize your data set, and you can use more functions also. Suppose you can create uh, PCA based, you can draw biplots also cluster based. There are various techniques available for creating this. So there are various packages also available in R, which you can use for PC and cluster. But this is a basic code for performing principal component analysis and cluster analysis. Okay. So this is from my side. Any questions? If you want to ask, you can please. Session is open for questions.